This lecture, I'm going to present to you some data that I've collected through my nonprofit. My nonprofit's called Channel Islands Cetacean Research Unit, and we collect data on dead stranded whales and dolphins to determine the health of the ocean through the health of the whales and dolphins. The first thing I want to mention is Earth is all connected. There are no habitats that are sequestered or independent from each other. All of our habitats are connected through the global water cycle. And so oceanic water is going to evaporate and precipitate up in the atmosphere, and then it will move over to the land where that water will precipitate down onto the mountains, and then that water is going to run off. And this cycle continues throughout every habitat, and it's connecting every habitat. Anything in this water is moving from one habitat to the next. So whether you're in the Arctic, the Antarctic, in California, in Europe, wherever you are not sequestered from whatever kind of industry or chemicals or anything that is anywhere on Earth, because all of that water is circulating on Earth, and it's all connected, and it all runs from these watersheds. So these are the different watersheds in the United States. And you can see here, this is the Mississippi watershed, obviously one of our biggest. We have the San Joaquin watershed over here, Central Valley. But what this is showing you is that wherever you are on Earth, whatever you put in the land is going to make its way to a watershed, and is going to make its way to a body of water, an ocean. And I think a lot of people have this theory like, oh, well, I live in Nebraska, I live in Iowa, I'm not connected to the ocean, or what I do on my land doesn't affect the environment, doesn't affect the ocean. But it's all connected, because whatever goes on the land is going to run out through those watersheds out to sea. And a lot of our watersheds are used for agriculture because it's water. It's water that's needed for growing crops or feeding animals or whatever. So we have a lot of agriculture around our watersheds, which drains a lot of different chemicals, pesticides, herbicides, whatever, animal feces, urine, any kind of diseases that are in animals are all going to end up in this watershed and run out to our oceans. And all of our oceans are connected. There is this huge current that runs all around all of our oceans. And this current's really important because it moderates the temperature of Earth. It helps cold water sink and warm water rise, and it moves water all through the planet. This is a very slow conveyor belt. It's not fast, but it is circulating water all throughout our entire planet. So again, whatever is in the water is circulating, whether it's trash, whether it's microplastics, whether it's nitrogen from fertilizing plants. Flame retardants are chemicals infused into textiles so that they don't go up in flames like this. Heavy metals from coal burning, pathogens from animals urinating and defecating near watersheds, noise and injury, all of these are having impacts on our oceans and our oceans animals. And we're seeing the effects of that. We're seeing mass die-offs, not just of mammals. We're seeing mass die-offs of fish. We had a sea star disease that killed out almost all of our sea stars. Brown pelicans have gone through episodes of high mortality. Mussels, clams, all different taxa are being affected by all of these different stressors that we're putting on them by dumping pesticides on lands, by using chemicals, by increasing our activity in the ocean, and by affecting our environment overall. Marine mammals are excellent sentinel species of their environments. For one, they feed at a high trophic level, so there's a lot of bioaccumulation going up the food chain, telling us about what are in those food, in those different chains, those different chemicals or whatever. Most marine mammals live in a coastal area, so close to humans, so we can see what we are feeding on reflected in what is accumulating in the marine mammals. They have abundant fat stores. A lot of the toxins that we're going to be talking about today are what we call lipid soluble, so they're going to store in the fat. A water soluble toxin will not store in fat, it'll just get passed out of the body. But anything lipid soluble is going to store in the fat. And then remember, these animals go through periods of fasting and feeding sometimes during the breeding season. Sometimes they're not eating, they're fasting, or vice versa. Sometimes during the winter they're fasting, depending on their habit. But during that time, they are relying on their body stores for feeding, for nutrition. And so they're going to be resequestering whatever chemicals are getting sequestered into their fats. They're going to be recirculating that into their bodies. And then finally, they're long-lived species. Some whales can live up to 150 years. So we can really get a huge lifespan, life history of these animals. We can really see what they're being exposed to over the different years. So my job as part of my nonprofit is to collect samples from these animals once they die. That's called a necropsy. An autopsy, auto is self. These are not humans, so we don't say autopsy. Necropsy is just the study of death. And it really can be as involved as really going through the animal in a detailed manner, collecting all sorts of samples. If the animal isn't very fresh, sometimes we don't get everything. We'll get different samples that are stable enough to withstand decomposition. And then sometimes, like a blue whale, requires extra equipment, and other times you can just do it right on the beach. Sorry for the gore. So each of these different parts of the animal are telling us different stories. We collect all different tissue samples to look for disease pathogens. We can look at the bone and the muscle to look for any injury, previous injury, or injury that may have caused death. We can collect toxins from the blubber. Like I said, a lot of the toxins are lipid soluble, so they're going to store in that blubber layer. Muscle, kidney, and liver can all tell us heavy metal concentrations. We can get biotoxins from any fluids in the body, urine, feces, semen, amniotic fluid, milk, whatever fluid is in the body, gastric fluid. And all of these samples are really telling us a lot about what's going on. So we're seeing animals that are still getting caught up with the fisheries and trash, debris. This is a gillnet. We haven't seen this in quite some time. The nearshore gillnet fisheries was banned in 1996, and that eliminated a lot of the animals getting stuck in the gillnets and dying like this. But we still get some netting strangulations. You can see this animal was completely wrapped up in rope, so this is probably associated with a crab pot or a lobster pot fisheries. We still see animals that are getting hit by ships. Sometimes they come to port draped over the bow of a ship, but other times you'll just see evidence. This flipper was cut off from the blades of the turbines and driving that ship, and then this animal had died from a ship, and you can see that its vertebral column, its backbone, was completely severed. All of this that you see there is all coagulated blood. That should not be free floating in the animal. This is not an open circulatory system. This is a whale. That should all be in a vessel somewhere. And then this is a rib bone that broke because of a ship strike. And you can see here at the margins of that break on that bone that it's this dark purple color. That's bruising, and that's an indication that this animal was alive at the time that it died, and its heart was still beating, and the blood was pumping or perfusing to these areas, and that's why you have bruising at the margins. If the animal was dead when this happened, you wouldn't have bruising at the breakage points on the bones. And all of these different causes of death and all of these data points are really showing us what's going on, and we're seeing lots of mortality associated with human-born or human-influenced causes. This here is a terrestrial-borne pathogen that washed down a watershed and caused a fungal disease. This is valley fever, San Joaquin Valley fever, coccidioides, and it causes these lesions in the lung like that. We're seeing cattle-borne diseases. We had killer whale, a newborn killer whale that was killed, that died because when it was born, its umbilicus separated from its body after it was born, and that umbilicus is an open hole or an open wound for a short period of time. And in that short period of time, the animal picked up salmonella, which was a terrestrial form of salmonella, and it killed that animal. So this animal died from salmonella that was in the water from cattle that had been feeding near a watershed. There's another disease called brucellosis, and so much brucellosis has ended up from cattle down watersheds into the ocean, and it's actually mutated into a form that we now call Brucella setae, which affects dolphins. So a lot of times people think as soon as diseases hit the ocean water, they'll die, but they don't always die. Sometimes they mutate and they survive. And if it's a novel disease, the dolphins haven't been exposed to it yet. They don't have any kind of antibodies or immunity to it, and it really can take its toll. We had a morbillivirus outbreak, which is from canine distemper virus. So a dog defecated on a beach, I think it was in Scotland, and that disease, that canine distemper virus, mutated and killed off hundreds and hundreds of seals in that area. Animals can also get parasites. This is a nematode or a parasitic worm that went into the ear canal of the dolphin and then migrated along the vagus nerve from the ear all the way through the brain, and it tunneled through the brain and caused death in this animal. And this is a protist, another type of organism, Sarcocystis, that was embedded in the heart muscle, the cardiac muscle of a blue whale. So lots of different parasites that can really wreak havoc on these animals. All right, let's start talking about some of the data that we're finding. So this data, we call these the persistent organic pollutants. These are the different toxins that store in the blubber tissue. These are what we're getting from blubber. And the persistent organic pollutants that I'm going to focus on, one is DDT. DDT is a pesticide that was banned in the 70s after Rachel Carson wrote a book called Silent Spring, how birds were dying because of eggshell thinning from DDT, and finally the EPA did something about that and banned DDT in the United States. DDT is still being used in other countries, especially countries with a high rate of malaria. Mosquito-borne diseases are one of the highest cause of death in some countries, in mostly third world countries in the tropics. And so DDT is still used to control mosquito populations. Mexico still uses DDT, so right south of the border, so it's still in our waters. And when there was an outbreak of Zika virus, DDT was brought back to try to control mosquito populations and get rid of that virus. PCBs is a chemical that's used in like transformers and power lines and things like that. It's an industrial chemical. And then PDBEs are the flame retardants. I showed you a picture of that chair on fire. So flame retardants are infused into every single textile, all of your clothing, all of your furniture, bedding, mattresses, all have PDBEs in them. And that's important because there's laws about, like, if you fall asleep smoking a cigarette or a candle gets too close to your clothing or your bedding, you don't want it to catch fire right away. And so the PDBEs will make sure that textile, that fabric will smolder for a period of time before it catches fire. But these are really persistent chemicals, and they're everywhere on Earth now. We're starting to find these chemicals in the Arctic, where obviously they've never sprayed DDT in the Arctic, but there are measurable levels of different contaminants up there. So a few things to look at when we look at this slide. The first thing I want to show you is that DDT is a very persistent chemical. It lasts a long time, but as it ages, as it weathers, it changes its form. We call that a different isomer. It's different shape. And so DDT can change into DDD and DDE. And if you look at the ratio of how much DDE to total DDT there is in an animal, it can tell you how long that DDT has been in the environment. And so generally we say anything 80%, 75 to 80% DDE or more is very old, very weathered DDT. And so you can see up here, this pie chart, we have 95% DDE in our common dolphin population in the Santa Barbara Channel. So pretty old, pretty weathered DDT. The good thing about that is some studies suggest that older, more weathered DDT doesn't have as much an effect on the immune system and the reproductive system that original DDT does. And so it's not as physiologically disruptive to animals. We collect samples from San Luis Obispo, Santa Barbara, and Ventura County. So San Luis Obispo is north, Santa Barbara in the middle, and Ventura is south. And you can see as you move from north to south, you get an increase in these toxin loads. There is a DDT dumping site off of Palos Verdes, California, and it's a super fun site. There's thousands of barrels of 55-gallon drums that have been dropped into the ocean as a way to dispose of DDT, and they're leaking. And so we have an active, super fun site of DDT leaking into the ocean. Again, it's older DDT. It's more of that DDE form, but it's still present. And this also shows you that as you get closer to population centers, you're going to have more influence of chemicals and toxins in the environment from those population centers. This graph is a little bit alarming. These are the chemicals, again, PCBs, the flame retardants, the PDBEs, and the DDT. And this is the concentration in females and males of the PCBs, females and males of the PDBEs, females and males of DDT. Why do you think they're so different? It's not because males eat more or eat in a different area. What's different between males and females? Females are giving birth. All of this concentration that is lacking in the females has been delivered to the fetus through the blood during gestation or to their calves through nursing. And we can see that here. Let's start down here. This is that same common dolphin data that we're looking at, but this data has been separated out. This is not paired data. So you see here mom, fetus, calf. They're not all related. This is based on the size of the animal that we sampled. So a mom would be anything over 180 centimeters would be a mother, would be an adult female. Anything from about 103 centimeters, 106 centimeters to about 160, 170 would be a calf. And then anything below about 106 centimeters would be a fetus. And we've sampled a lot of animals, and what we have found is when you compare the concentrations of PCBs, PDBEs, and DDTs, you can see moms have pretty low levels because they are dumping their levels into their fetus and into their calves through nursing. This data up here is actually paired data. This was a blue whale that died from one of those ship strikes, the one that had her flipper cut off, her fin, and the mother was pregnant with a fetus. The fetus was about 13 feet long, so it was about halfway through gestation. They're born about 25 feet, the blue whale calf. I know, huge, huh? And so even halfway through gestation, we sampled that calf. And that calf has measurable levels of DDT, PCBs, and some other lindanes and chlorines or other chemicals that we're sampling for as well. So this fetus is starting out its life with already being toxic. And some of these toxins have shown to disrupt estrogen and testosterone, so to disrupt reproduction, to disrupt the immune system, to disrupt hormones. So everything physiologically about these calves that are being born with these toxins are challenged from birth. They're already challenged at birth. Remember, they don't have much of an immune system at birth. They need to develop that over time. And so to already start out life being compromised is such a challenge for these animals. These are some of the heavy metal data that we're looking at, and this I just separated out mercury for you. Remember, this is coming from coal burning. And when you sample the different animals and you look at cause of death, you can see that animals that die from illness have a much higher level of mercury in their liver than any other cause of death. What is also alarming is that mercury continually increases in the animal as the animal ages. And so as an animal gets bigger, its concentration of mercury goes up. And we are almost to the point where we can age an animal based on how much mercury they have in their liver. How sad is that? During my studies, a really interesting opportunity arose, and a colleague of mine is studying a part of the animal. It's called the ear plug. It's wax that builds up in the ear canal of baleen whales. Only baleen whales get this. And you can actually separate out the layers of this ear plug, and it can tell you how old an animal is, or can tell you different things that happen throughout its life. And a colleague of mine, a friend, actually developed this study to separate out these lamina and compare some of the metrics that we have already talked about in each of these layers to see how much. So we were looking at total body composition of DDT and mercury and that kind of stuff. He wanted to see how much that animal accumulated every year of its life. So here's the external ear canal this is a humpback, and that here's the ear bone in purple, and that ear plug would be in this kind of groove here. Again, here is that ear, the ear bone, here's that ear canal, and this is where the wax builds up, and this is what ours looked like. And then this one, you can actually see the different layers of it. And so what he did was he separated out these layers and looked for DDT, PCB, PDBE, mercury, testosterone, and cortisol from all of these different layers. If you remember when we talked about mating, I showed you the testosterone and cortisol. This is where this data came from. You notice that some of these layers are dark red and some of them are kind of whitish. This is because blue whales spend half of their life, half of their seasons, half of their year feeding on large amounts of krill. All summer long, they are feeding, trying to bulk up on as much food as they can, and that's all krill. And that leaves this red line. It stains their fat red. During the winter, the animals go down south to warmer areas where there's not a lot of food, and they're breeding. And so they are relying on their blubber stores that they stored up when they were feeding for nutrition while they are in the breeding grounds. And that is that lighter layer here that you can see in white. So let's take a look at what the data was showing us from the earplug. First, let's look at some of the persistent organic pollutants, that DDT and the PCBs. And what you see here is this is age along the x-axis, and then this is the concentration of the different chemicals. These animals have very high concentrations in that first layer of that earplug, that first lamina. These animals are born with very high concentrations of DDT and PCBs. In fact, 20% of their total body burden was acquired in their first year of life, their first 6 months of life. That's alarming. And then for this animal, it was 80% DDE, which again, that's considered an older form of the DDT. You can also notice you have these kind of peaks and valleys. That is because, remember, these animals are feeding, fasting, and so they're going to pick up more contaminants while they're feeding and less when they're fasting. They don't pick up zero when they're fasting because they're recirculating their blubber stores that already have measurable concentrations of these contaminants. We see the same thing for the flame retardants. They are born with about 20% of their whole body burden, and then you can see those same patterns of feeding and fasting. This is part of a validation study, and it actually helped us because he also did the same analysis from a gray whale that was much older than this animal, and the earplug had been sitting at the Smithsonian Institute in Washington, D.C. And what he found was that there was no PDBEs prior to 1969, which makes sense because they weren't really being used before the 70s. And so it's validating that what is in these earplugs is in the environment at the time they are picking it up and is in use at that time. So it's very validating. What's also interesting is I put this red line here. This is the total body burden that we measured in the blubber. But if you look at what the animal had accumulated over its lifetime, that's this black line here, and it's actually at the time of death higher than its total body burden. And that's because the animal, again, has a higher level of these toxins in its earplug because it's mobilizing those contaminants when it's fasting. And so it's re-ingesting the contaminants that it had already laid down in its blubber layer. So the blubber layer is slightly less than the amount of contamination in the earplug. The mercury levels were a little bit intriguing. First of all, they don't follow the same pattern that we see in the other contaminants, the persistent organic pollutants. What we think happened is that there was El Nino and La Nina years when this animal was alive, and so during El Nino years, when there's more storms, some of that mercury gets resuspended into the water and picks it up more, and then other years there's less. But what's most alarming to me was the liver mercury of this animal was 1.9 parts per million, and the amount in the earplug was 340 parts per million. So I feel like we're missing a lot of the mercury in the liver than compared to what's actually in the body. So after this, we started looking at methyl mercury because that's kind of the form that it gets broken down to in the liver, and that's giving us some more exact data. You've seen some of this data before. The cortisol, that stress hormone, remember it peaks at the same time that testosterone peaks. We've talked about this because mating is stressful. Why do we care, and what does this have to do with us? Well, the first thing is that these animals are feeding on fish in the same area where we're fishing and we're getting our fish from. So whatever is in their diet is in our diet if you're feeding on fish. Also, these animals are very long-lived, so they're really bioaccumulating all of these contaminants over a long period of time. There are also some populations that actually feed on whale. There's a subsistence bowhead whale hunt for the Inuits in Alaska, and they actually feed on all of these different parts. They're feeding on the blubber, they're feeding on the mussel, they're using all of the different parts in their daily diet and in their daily lives. And you can see when you compare the Inuits that feed on the subsistence whale compared to the Inuits that mostly feed on caribou, look at the contaminations in their bodies. These are humans. These are PCBs, DDEs, and the H and other contaminants, the chlorhexidines and things like that. But you can see there are very high contaminants in humans that are feeding on these animals. Other populations feed on whales too, the Japanese, Norwegians, so it's not just the Inuits that are feeding on them. I know this one's hard to see, but it's such a huge laundry list of the effects of pollution. It affects every part of the body. It affects the respiratory tract, the cardiovascular tract, the endocrine system, the neurological system. It's a carcinogen. It causes cancers. It affects the gastrointestinal tract. Multiple ways it's affecting all of these systems. And hopefully by now you see how integrated these systems are in keeping animals alive. And if you are completely bombarding these systems constantly with contaminants, it's making those physiological systems, keeping that animal alive, so much more challenging. And overall, it's going to lead to premature death. Again, the human health implications. We are eating fish. There is mercury in the fish that the animals are eating. There are contaminants in the fish. This Monterey Bay Seafood Watch program is great. It kind of gives you more sustainable fish to feed on and fish to avoid that are not sustainably caught, but it doesn't take into account how much contamination are in those fish. And some of these fish that are on the best choices list are highly contaminated fish. So again, you need to really think about what you're putting in your mouth. Now let me caveat that by saying these animals are only feeding on fish. That's all they feed on. And I know that there are no humans out there where the only thing they eat day and night, every meal, is fish. So you're not accumulating the amount of concentrations that these animals are getting. We eat a more varied diet, but it should start to get you thinking about what you're feeding on. Are you feeding on organic food? Are you eating locally grown grass-fed meats, pasture-raised foods? Because whatever is going into the food is getting into our bodies. The other concern too is the synergistic effect of all of this. When you have noise pollution from all of the activity, and then you have these contaminants in the body and the heavy metals and biotoxins, which we didn't talk about but are also another problem, and parasites and other diseases, 1+1 doesn't always equal 2. And we're starting to see that now. I showed you the data with the bottlenose dolphins from after the Deepwater Horizon. Animals are starting to stress to death. There is just too many stressors on them. They can't survive, and we're starting to see mass mortality events. We're starting to see higher rates of mortality, lower body weight, lower reproductive rates in these animals because they are just carrying too many burdens. One thing you can start to think about is your ecological footprint. How are you affecting your world around you? How much are you using from your world? So you can start to think about your life and what you're doing. We've talked about better choices. Those K-pods are the worst. Single-use water bottles, you don't need them. Single-use shopping bags, you don't need those either. So if you can eliminate a few things in your life to make the stressors a little bit less on animals, it would be worth it. All of this plastic comes from oil. If you have an issue with big oil, your first line of defense is to get rid of plastic in your life. And not only is it coming from big oil, but a lot of these different pieces and parts of them get manufactured in China, get shipped over here for different things. Sometimes they go back to China and then come back to the United States. Sometimes these bags and things can have multiple transits across the oceans just in the different manufacturing parts. And all of that is creating noise and injury for our animals. So it's all additive, it's all encompassing, and it's all related, and it's all connected. Remember that everything in this planet is connected. There is no sequestering these contaminants. You can't say, oh, well, China manufactures all those chemicals, so all the chemicals stay there. That's not how it is. Or, oh, those DDTs being used in the South Pacific, we don't have to worry about it. That's not how it is. It's all moving around the Earth. The entire ecosystem of Earth, they're all connected. Nothing is in a vacuum. And so it's really important to start to consider our actions